Okay, what we're going to talk about today is the CO2 transport from tissue. We know that as metabolism undergoes, we start making waste products. Those waste products can accumulate nitrogenous waste, phosphate, water, CO2. Specifically, when we break down carbohydrates, we're going to make a lot of CO2. CO2 is a problem. All of these waste products are essentially a problem. We have to get rid of them, and the body has to clear them. So this simple schematic, what we're going to look at here is these blue cells here are going to represent tissue cells. Okay, and inside the tissue, the one particular waste product we're going to focus on today is CO2. Okay, let's put a couple CO2 molecules in these cells. Right. So these CO2 molecules, they got to go. Metabolism is complete. We've used all the nutrients and things that we wanted to use, and our body has made some waste product. Now, these waste products, they gotta go. So, how do they go? Well, we have to transport them, and we have to transport them through the blood so that they can get to the pulmonary circulation, to the alveoli, and finally exhale them. So, how does that all happen? Well, there's essentially three different mechanisms that play a role in CO2 transport from the tissue. The first one is something just called dissolved CO2. The second one we're going to talk about is the bicarbonate anion. And lastly, we will talk about something called a carbamino compound. All right, we'll see if we can fit this all in one screen. So first off, let's talk about this dissolved form. Well, this is actually the simplest form of CO2 clearance. So let's say this CO2 molecule right here, okay? Based on simple diffusion, this concentration really high in the tissue cells, it diffuses into the plasma and it stays in its dissolved form, okay? So it just transports right down the circulation to the pulmonary circulation and back out to the alveolar where it's exhaled. Now it seems like a great way to do it, simple, stays in one form, but this is actually a very small percentage of how CO2 is transported. This is roughly around 8% of how CO2 gets transported through our body. Reason being, this has to dictate based on Henry's law. This is partial pressure, so if things are constant, diffusion rates. Oddly enough, though, this is, as respiratory therapists, this is what we look at when we do an arterial blood gas. So when we read our PaCO2, we're reading this amount of dissolved CO2 in our blood. Okay, we're not reading about the other mechanisms. Indirectly, we can get an idea, but this is particularly the one that the sensors are going to look at, or the electrodes are going to look at when we analyze an ABG. So that's the simplest form. That's your dissolved. Okay, small amount, only around 8%. Stays in this form, exported down, exhaled out. Okay. By far and away, the largest way we could transport CO2 is through this bicarbonate anion. Now, what happens here is, like we said, this waste product diffuses out into the plasma and then gets right into the red blood cell. So if we haven't figured this out yet, this red tube-like structure, say it's a capillary at the tissue, and this big circle here, that's going to represent our erythrocyte or our red blood cell. So let's start putting in some of these CO2s. Okay. This CO2 now reacts with other things, other waste products even, such as water. And these are both now inside the red blood cell. This CO2 and water combine to form something called carbonic acid, H2CO3, carbonic acid. That carbonic acid further dissociates now into a proton and a bicarbonate anion. Okay. Now, this is called our hydrolysis equation. And this happens within the red blood cell. And it actually happens very, very quickly because of the presence of something called carbonic anhydrase. This carbonic anhydrase is a catalyst, or it's an enzyme that helps speed this reaction up in either direction. Okay? And that speed is going to help produce a lot of bicarb. Let me grab a different color here. Grab green. This bicarb then, because of the speed of the reaction, the hydrolysis reaction based on this catalyst, is going to produce a lot of bicarb. So all these molecules, or 80% of these molecules, can come in and get dealt with through this mechanism, and we can transport a lot of CO2 in form of bicarb. And it's one of the main purposes is because of this assistance of carbonic anhydrase. So this anion is going to start to accumulate within the RBC, and the RBC is not going to want that. It's going to start saying, you got to get out of here. We got to move you along so that we can do something more beneficial for the inside of this RBC. So what happens is this bicarb anion simply just swaps out into the plasma. So there goes the bicarb from inside the RBC out into the plasma. Now that it's in the plasma, it's left a deficit of a negative charge, or an anionic charge, inside that RBC. And the RBC likes to be balanced. It likes to have electroneutrality or homeostasis. So it needs to get that negative charge back. Otherwise, it's staying in this cationic or positive charge. It's at a deficit right now. Well, luckily, one of the more abundant anions that floats around in the plasma is this thing called chloride. And the chloride then shifts in as the bicarb shifts out, and both of them, or the charges, start to equal. One negative out, one negative in. And now the RBC on the inside maintains that electroneutrality. But we do have to do something with this guy right here, this proton. Let me grab another color here. So this proton right here, they're kind of nasty guys when they're left free to do their work. These can cause changes in pH. So this left unattended is going to cause a change, an internal change in the environment within inside this RBC, right? And the RBC doesn't want that. So something has to buffer this. It has to decrease the change or the effect of the acidity of that molecule. And we know that inside of an RBC that we also transport oxygen. One of the main functions of the RBC is to carry this substance called hemoglobin. And hemoglobin carries oxygen. And we're going to represent an oxygenated hemoglobin by this symbol here, HbO2. So that would be an oxygenated hemoglobin that then can offload this oxygen to a tissue cell where more metabolism then can continue on. But there's also deoxygenated hemoglobin molecules. And those deoxygenated hemoglobins can offer up their service to buffer out things like that proton. So that Hb deoxygenated or anion can bind with that proton and form a protonated hemoglobin molecule. And now that protonated hemoglobin molecule helps decrease the change to pH in the internal environment. Okay? So all of those things kind of happen because the bicarb swapped out, because this was in such a high concentration due to the action of carbonic anhydrase, which produces lots and lots of these products in such a high speed or high rate, that the inside of the cell says, you got to go, you got to get out. So it kicks these bicarbs out. The bicarb then, which is representing, really, it's representing our CO2 molecule, because if we trace our steps again, that CO2 molecule dissolves in, gets into the RBC, combines and makes carbonic acid, and then dissociates into this bicarb. That bicarb gets kicked out, and that is essentially now representing a lot of the CO2 molecules that came from our tissue. They're going to get transported now to the pulmonary circulation. But when we did that, chloride molecules swapped in to maintain the electric neutrality, and then we said this is called the chloride shift. Okay, so you'll see that in textbooks called the chloride shift, or even some textbooks refer to it as the hamburger phenomenon. This left that proton to kind of change some of the acidity level of the pH inside the environment, so we used the deoxygenated hemoglobin molecule to buffer out that change. So hopefully now everything is happy. So now we've got about 8% of our CO2 taken care of in this dissolved form, and we actually have about 80% taken care of in this bicarb form. So that's a very substantial amount of CO2 transport in the form of bicarbonate. Okay. And then the last formation of transportation of CO2 is something called the carbamino compound. And the carbaminohemoglobin is the final transport mechanism, we'll say, where CO2 binds with an oxygenated hemoglobin molecule. And this is due to the affinity of hemoglobin for CO2 and the abundance of it. This competitive binding, for instance, works in favor at the tissue because when the CO2 binds with hemoglobin, it knocks off an oxygen, which is already needed to be delivered to the tissue. So let's see if we can kind of do that in a picture here. Let's grab another color. Let's try this one. All right. So we'll say a CO2 molecule gets into let's grab this guy here. So we'll say he represents this CO2 molecule. It is then going to combine with, well, we already know there's this oxygenated hemoglobin in here, right? See, we didn't do anything before, but it's going to help us out now. So this carbamino compound, we're going to make from a CO2 molecule and this oxygenated hemoglobin. So this then is going to combine to make something. We're going to draw it a little bit different here. We're going to draw this carbamino hemoglobin as an HB, and we're going to put a CO2. We're going to just make it look a little different like that. So CO2. Okay. We're going to combine that with oxygen, as per right there. So there's an oxygen molecule, and there is usually some sort of proton associated with this as well. Okay. We don't have to worry too much right now about that. We talked a lot about how that buffering capacity can be used for protonated hemoglobin. Okay, so this can be used as well. Now, what this does, this molecule needs to get up to the tissue. That's the whole purpose of internal respiration. We need that oxygen molecule to get up into the tissue so that we can utilize it, make ATP, undergo cellular metabolism and respiration. That's perfect because we needed that to get up into the tissue, and now we can carry this CO2 molecule on this carbamino compound and bring it all the way back to the pulmonary circulation to offload this CO2 and then pick up another O2. And by picking up another O2, it'll knock that CO2 molecule off, and this just keeps going around and around and around. So this carbamino compound represents roughly 12% of how CO2 is transported in our blood. And that's the final mechanism. Hopefully, it looks kind of busy as we look at the screen now, but hopefully that kind of is a good, or serves as a good review of some of the ways CO2 is transported from the tissue to the blood. And then you guys, if you want, you can flip these roles and try to figure out how from the blood this goes back into the cell, and the hydrolysis equation starts going that way, leaving a CO2 molecule to get freed so that it can get exhaled out. All right, guys, hope you enjoyed the little webcast. If you want to see anything else throughout the course, please don't hesitate to let me know. We'll try to make things come to a visual way that we can see it all. Thanks. Have a good day.